Good afternoon, everybody, and welcome to the Department of Psychology's annual Quinn Memorial Lecture. My name is Jeff Hall. I'm the department head. Before we begin, I'd like to take a second to acknowledge that UBC is located on the traditional, ancestral, and unceded territory of the Musqueam people, and we thank the Musqueam Nation for its hospitality and support of our work. This year we mark the 12th anniversary of the Quinn Memorial Lecture, and this is an event that's made possible by a generous gift to our department from our alumnus, Dr. Michael Quinn. In 1969, Dr. Quinn became only the second person at UBC to obtain a PhD in psychology. After he received his doctorate, Dr. Quinn went on to have a long and distinguished career as a clinical psychologist at Riverview Hospital in Coquitlam. And in addition to this distinguished lecture series, Dr. Quinn's endowment has allowed the Department of Psychology to establish many other important initiatives, including a number of important undergraduate and graduate student research and travel awards. And tonight, we're especially honored to have Dr. Quinn's niece, Mrs. Agnes Douglas, and his great-niece, Ms. Wendy Sutherland, with us here in the audience. And so we're just very happy that you could be here tonight to see firsthand the impact of Michael's generous gift. This year, we are delighted to welcome Dr. Robert Knight, Professor of Psychology and Neuroscience at UC Berkeley. Dr. Knight holds a Bachelor of Science degree in Physics from the Illinois Institute of Technology and an MD from Northwestern Medical School. He was on the neurology faculty at UC Davis for many years before moving to Berkeley in 1998. Dr. Knight is a preeminent cognitive neuropsychologist renowned for his groundbreaking work using brain imaging techniques to study the contribution of prefrontal cortex to human behavior. His research interests include attention and memory, neuropsychology and physiology, and cognitive neuroscience. His laboratory uses electrophysiological, fMRI, and behavioral techniques to study controls and neurological patients with frontal lobe damage, trying to understand the neural mechanisms underlying cognitive processing in humans. Dr. Knight's list of commendations is as long as his distinguished career. He has twice received the Jacob Javits Award from the National Institute of Neurological Disorders and Stroke. He has also received the IBM Cognitive Computing Award, the German Humboldt Prize in Neurobiology, and the Distinguished Career Contribution Award from the Cognitive Neuroscience Society. He is a Fellow of the American Association for the Advancement of Science and a member of the American Academy of Arts and Sciences. And tonight, Dr. Knight will share recent discoveries from his research program in a talk entitled Insights into Human Cognition from Direct Brain Recording. So please join me in welcoming Dr. Robert Knight.
I wanted to say A. Does that sound right? I have one of your PhD students in my lab as a postdoc, Julia Cam, who she says A a lot, so must mean she's Canadian. So just a couple things. This is Helen Wills, and Helen Wills I want to make a point about what philanthropy can do for science. She was a Berkeley undergrad, and then she went on to win Wimbledon 8 straight times, never lost a set. Between 1925 and 1933, she had a very great life, married well, and left her entire estate to form the Helen Wills Neuroscience Institute, which is really drives an incredible span of research at UC Berkeley. So we're very, very indebted to Helen Wills Moody. Martina Navratilova did break her 8-win record with 9 wins, but Martina Navratilova did lose multiple sets. And the reason I think is the brachioradialis muscle. I hope you can see there. She had a killer backhand. Anyway, I'm particularly interested in the frontal lobe and how it regulates behavior, how it allows you to decide to come here, how you're paying attention. The story goes back to 1876 when the British neuropsychologist David, am I on? Is this one on? Which one's on? Do the lapel because I don't want to lean over. Is it good now? Can you hear me in the back? I can't tell. Can you guys hear? Okay, good. So David Ferrier took what he said were his 3 smartest monkeys. By the way, this gentleman is the person who discovered visual cortex. Took off their frontal lobe and observed their behavior. And he couldn't identify any specific deficit other than the monkeys seemed disinterested in the environment. This led to subsequent work many years later by Alexander Romanovich Luria, who I think is probably the most brilliant neuropsychologist ever, Russian in the lineage of Sokolov. And he, from observing patients with frontal damage, came up with these ideas about the frontal lobe. The frontal lobes are essential for organized intellectual activity as a whole, including the programming of the intellectual act and the checking of its performance. Now, this is based on observation. We're now 50 years later, and abundant, very expensive imaging techniques have really simply confirmed his initial incredible observations. Now, why am I interested in the frontal lobe? I'm a clinical neurologist, in addition to being in a psychology and neuroscience program. And frontal lobe damage is ubiquitous, and it's very, very common in both neurological and psychiatric disorders. So if you just look here, here's examples of someone who has a contusion in the frontal lobe from falling down some stairs. There's chronic loss of tissue in the frontal lobe. Here's an acute fatal injury. Here's a brain tumor pushing up on the orbital frontal lobe. All these patients tend to have problems with social regulation, orbital frontal damage. Lateral frontal damage, the most common is probably stroke. Here's 2 examples of stroke. Here's an infiltrating brain tumor, the glioma tumor, which is horrible because it's just a bad tumor. You can delay the final outcome, but not for a long time. You have degenerative disorders. You can see this very severe atrophy of the frontal lobe. This is another form of dementia. It's not Alzheimer's. It's called frontotemporal dementia. And you can even have cortical malformations, of which this talk is going to be focused on epilepsy patients, where we're trying to find the cortical malformation and take it out to stop the seizures. So you could replace this slide and say psychiatric disorders, and you'd have schizophrenia with lateral frontal damage, obsessive compulsive disorder with orbital frontal changes, and so on. So understanding how this brain region regulates behavior is very important. So here's Ferrier saying the monkeys, instead of being actively interested in their surroundings, they were apathetic or dozed off, responding only to the sensations or impressions of the moment. They had lost the faculty of attentive and intelligent observation. Here's a human with a bilateral frontal lesion, and you'll see he's apathetic, disinterested, and he shows these primitive release signs, these signs that your babies have, where they grasp things and they go for the nipple and go to suck things. They all return when you lose your frontal lobe. Okay, don't do. Okay, there's a bilateral grasp reflex. Okay, like your baby, both hands. Okay. Let me just check some things. Watch his lips here. Purse. I'm going to tap you. It's not going to hurt. I'm just going to give you a little tap. See his lips purse? Can you see that pursing? It's a classic frontal lobe sign.
I think you can see there's a
patient, something's not right with the frontal lobe.
You say 4+ snout reflex. No clear rooting reflex.
Lesions.
Open your eyes. Open your eyes, sir. Open your eyes.
You see he's profoundly under-aroused. Why is he under-aroused? He's under-aroused because the projections from the frontal lobe to the rest of the brain are key to regulating those areas and establishing the proper excitatory balance. When you look at the back of the brain in this patient, it doesn't work. It doesn't work because of this loss of control from the frontal lobe. How about if we flip the brain and look at the bottom of the frontal lobe, the orbital frontal part of the cortex? And this is the kind of damage you see in the classic, which any of the psychology people know, Phineas Gage, the person who had a rod go through his orbital frontal lobe and had a profound change in behavior, but remained intelligent. Here's a gentleman who ran his scooter into a motor pole and completely took out his entire orbital frontal cortex And you'll notice he's completely different. He has a chaotic lifestyle, he can't maintain relationships, and he can't read people. He's lost the ability to understand social behavior.
Did you notice that you were attracted to a lot of women or just not attracted? What was it?
I was attracted, very much so.
To a lot of women?
Yeah. I touched things 4 times,
Acquired OCD, very common.
put things in my mouth. Everything's an even number. All my 4s, 8s, or 16s. I count license plates, I count flags.
What's the problem that you're not what you thought you would be prior to the accident? Something is impeding that, obviously.
I don't have a whole lot of concentration.
IQ is 130, by the way.
I don't like being told what to do. I've had trouble maintaining a job ever since the accident.
Chaotic
I am not
lifestyle.
that perceptive. I'm naive.
People can still take advantage of you on the streets?
I believe people.
So you haven't had any fights, you haven't been arrested in the past 20 years?
Oh yeah, many times.
What for?
Twice on assault against my wife. Although, like I said, I need to replace the door. The only thing that got beat up was the door. And for improperly using somebody else's credit card. And I wanted to open a checking account and bounce $2,000 in checks in a week.
Was that the right thing to do?
No, of course not.
Any other arrests?
I had a DUI, 2 DUIs, and a reckless. One in Hawaii, one in Washington.
This patient, if you just see him in the clinic, is often diagnosed as a borderline personality disorder. He's not a borderline personality disorder. That's the result of him damaging his orbital frontal cortex. We'd all have the same behavioral problem. God forbid it happened to you. So my lab is really trying to understand what's going on physiologically, and we do a lot of research in brain lesion patients, but I'm not going to talk to you about any of that today. I just wanted to frame the problem. What we're going to talk about is direct brain recording. The electrical signals of your brain were discovered in 1929 by Hans Berger, a psychiatrist who was trying to develop a method to distinguish different kinds of psychiatric disorders, and through some elegant studies, including new amplifiers, he showed that the brain signals that you recorded on your scalp were coming from your cerebral cortex. Major discovery. What we're going to do is decompose these brain signals into multiple frequencies. What does that mean? If you go to a jazz club and there's a bass player, there's a piano player, and there's a saxophone player, they have 3 different center frequencies. We can use a technique called Fourier analysis to decompose the rhythm in the jazz club into power in the low frequency bass, into the higher frequency sax, and even into the piano. It's just a spectral decomposition. So I'm going to show you, it's a little bit more complicated than that, but not a lot more complicated. We're going to take your brain rhythms and decompose them into different frequency bands. Now, it used to be felt that your brain only generated electrical activity up to 50 hertz. That's 50 cycles per second. And this really held until 2000, when a colleague of mine at Johns Hopkins, Nathan Krohn, in one patient discovered that there was abundant high-frequency activity to phoneme stimuli in the 65 to 200 and 250 hertz range. It turns out this is the most important rhythm in your brain. This is what's tracking everything you do. As you hear, your auditory ears are decoding sounds using this in this frequency band. As I decide to pay attention over here, my frontal lobe is controlling my eyes so that I look over here. And if I look directly on the brain, that's the rhythm. Now you say, but I can't record it on the scalp. It's too small amplitude. I have to be directly on the brain. So how do we do that? Well, unfortunately, 1% of the population, including 1% of the population of Canada, has epilepsy. It's a very common disorder. How many people in this room know somebody personally or a friend who has epilepsy? Look around the room. Just take a peek. Right? So this is not an uncommon problem. Mercifully, 85% of them are controlled by medication, but 15% are not. If we can localize where their seizure onsets, we can take it out and get control of their epilepsy. So I'm going to show you data from patients who have uncontrolled seizures. We bring them into the hospital. We implant electrodes either on the surface of their brain, if that's where we think the source is, or deep in their brain in different key structures that the psychologists would know: the memory area of the brain, the hippocampus, the social area of the brain, and the orbital frontal cortex, the kind of control, checking your behavior area in the cingulate. And you can see these electrodes. You say, God, that's horrible, but they're really very fine electrodes, and just a little teeny hole in the skull, and we insert them robotically so that we know precisely where we want to put them. The patient sits in the hospital for 4 to 10 days until they have 3 clear stereotype seizures where we know the onset of the seizure and they do the behavior they typically have. As soon as we have 3 seizures, we stop, they go to surgery, we take that area out, and we get very good control in the brunt of patients. So I'm going to show you data while the patients are in the hospital. If they want to participate in research, they can. And luckily, most patients kind of know we're not doing this for fun. We're trying to figure out how the brain works, and although it's not directly helping them, that it might help somebody else. So before I start showing you some of the data, I wanted to show for the psychologists in the audience that we're also beginning to record these electrode shafts that we put in. We can put little wires at the end that allow us to actually record single unit activity, the actual firing rate of an individual brain cell. And this is really remarkable. And you can see here's an electrode in the patient's hippocampus, and I don't know if you can make it out. Those are the little fine wires, and these are 2 separate types of brain cells firing from one of those wires. So that's coming. We're just getting started with this. We kind of don't know exactly what we're doing, but we'll figure it out. We can take the data from patients and we can put it together in atlases across the patient population. This is some nice work by Arjen Stoke from the Netherlands. And this is the sum of many patients. We can have a localized all the patients who have electrodes in the orbital area, all the ones who have electrodes in the motor area, and so on. So we have a lot of control of the anatomy and the physiology and the behavior. The 3 key things you need to understand how you do things: anatomy, physiology, behavior. So I'm going to show you 3 lines of evidence in this regard. First, I'm going to show you evidence that this high frequency band, this 70 to 200 hertz band, tracks every behavior you participate in. Second, I'm going to show you that your brain functions not in a spot, it functions in a network. Right? Phrenology, that love is here and hate is there, it's not true. We have network activity, and I'm going to show you that coherence between 2 brain areas allows them to be in sync and rapidly transmit information in the sub-second domain, which is how we're functioning, by the way. Everything we're doing is a sub-second. We're fast. We're fast. You see artificial intelligence, it's going to replace the brain. Remember, they've aptly named artificial intelligence. It's artificial. It's not human intelligence. But any of you out there that I've offended, tough. Tough. I didn't mean that. That sounded American. I'm really sorry. I apologize. Is that right? Apologize? Okay, I'm okay. Fine. Okay, sorry. So, this is like a patient that's been, has electrodes in. I'm going to show you that long-range coherence selects rapidly networks in the brain. I'm going to show you that the slow oscillations in your brain tune when high-frequency activity occurs. So we've got a tuning mechanism. And the tuning mechanism really is very powerful because it allows you to have things happen at precise time intervals. And you can just see here again, that's what we're going for. Okay, so let me show you first a real simple study. This is work by Eddie Chang, who was a postdoc in the lab, fabulous. It's a simple working memory study. First you just hear a bunch of phonemes, I, E, A, U. Then you have to detect whenever you hear a U. No big deal. It's pretty simple. A, I, E, A, U. Press a button. Okay, that's the target. Now you make it a little bit harder, where you have to hold in mind the sequence of events that happened, because you're now going to be asked to press a button if and only if U occurred 2 stimuli before. It seems really easy. It's not really easy. It requires you to keep a running track of the recent past for comparison to the present activity. So what happens in your brain if I record a patient with these electrodes on the key areas that are involved in working memory? I'm going to show you, these movies are 2-second movies, and they're spread over 10 seconds or they go by too quick. So here's where you just have to detect an ooh, here. This is going to show you when you have to detect an ooh only if an ooh occurred 2 stimuli previously. Okay, so watch what happens here, and I'll see if I can stop this at the right time. I'll show you the maximal activity. Just went by it, whoops. Okay, let me back this up. I'm not too good at this. There. That's the maximum activity in the frontal lobe in this task, in this relatively simple task. I want you to watch what happens in the frontal lobe when I give them the more difficult task. You're going to see a dramatic increase in activity. Oops. I think you probably saw it. I just screwed up. You're also going to see, as this unfolds, massive increase. You're going to hear the ooh, and then you're going to get this massive increase in frontal activity, and then you're going to press the button. That's your hand representation, pressing the button. So we're able to track the behavior in this simple task using this high-frequency brain activity, and you show clearly that the frontal lobe implicated in working memory has increased high-frequency activity. What about language? Everybody, maybe, I shouldn't say that, most people have heard of Paul Broca. Paul Broca is reported Broca's aphasia, which is a stroke in the frontal part of your brain where the patient understands what they want to say, but they can't say it. So listen to this gentleman.
Let's check your language out now. Can you tell me what happened when you had the stroke?
Ja, sitting this happened. Sorry.
Do you remember, was it in the morning or the afternoon?
TV,
He knows what he
TV.
wants to say. He can't
11?
get the
11
words
in
out.
the morning?
Very frustrating.
11 in the morning. Okay, so he's understanding what I'm asking him, but he has a non-fluent output of speech, sometimes just single utterances, but he's trying to
He's got right
communicate
arm
the
weakness
correct
also.
content. He knows the question. He's showing me it's 11 a.m. Can you tell me, where were you born?
63, 68.
63, 66.
60.
I see, okay. Where were you born? What city?
Windland.
Where?
Windland. Linwood.
Linwood?
No, Windland.
Let's just try some
You can
simple
imagine how
language.
frustrating
What's
this
this
is.
called?
When you know what you want to say,
To
and you
be.
can't communicate.
To be, yes. To be.
Can't name it, knows
It's
what
kind
it
of
is,
a phonemic
right?
error for
Obviously.
toothbrush. He certainly knows what it is.
Okay, so two
so-
things about this patient. One, at the end of this talk, I'm going to show you what we're working on to develop a speech decoder so that when the patient wants to say what he wants to say, we try to decode directly from his brain his thoughts and play it on a speaker. So I want you to hold that in your mind's eye in your working memory till the end of the talk. The second thing is, if you ask a room of how many people in this room think that the psychologists think Broca's area is active when you're speaking? You guys don't have any opinion? Yeah, I mean, that's the party line, is that Broca's area is involved in speaking. Well, guess what? It's not true. And that's over 150 years. This is my friend Nina Dronker's colleague, and she's holding Broca's brain. She's very excited because she's a neurolinguist and she has the classic brain of all time in language. And she's going to take it and we're going to float it in an MRI scanner and actually do a scan of the brain. And which we did. They let us have it. It's in the big anthropological museum in Paris. There were actually two of his brains there. Two cases. This is the first one, and we were able to get this one open. The other one, we couldn't break the wax seal, and they wouldn't let us break the glass, so we never got to scan it. So anyway, When you scan Broca's brain, what you find, yeah, Broca's area is damaged, but so is the underlying insula and arcuate fasciculus. And I apologize to people who are not neuroscientists. That suggests there's more damage than just Broca's area here. Second, if you take a cohort of we have a large stroke cohort with language problems. This is, I think, 36 or 37 patients with a chronic Broca's aphasia, and we overlap their brain lesions. Actually, the center of the damage is not in Broca's area. It's actually deep in the insula and the arcuate fasciculus. So this is a problem for classic theory of language. It just doesn't fit. So how about if we do this direct brain recording and figure out what's going on? So first, this is some work by Eric Edwards. If you do an fMRI scan, so the smart guy, I guess, is the chair, right? You're the chair, right? Okay, so you have to do the task. I'm very sorry. But of course, as the chair, you could assign it to another faculty member, but that would be abusive. That is, I didn't think you did abusive things in Canada. I thought that was only Donald Trump who did that. No, here's what I want you to do. I want you to do what's called the verb generation task. It allows us to track the language area. All I'm going to do is give you a noun, and I want you, she's getting nervous. By the way, undergrads do this in 1.3 seconds. And I'm timing you. So no, seriously. And I'm going to give you a noun, and all I want you to do is to generate an action verb to that noun as quick as you can. Okay? Ball.
Throw.
Perfect. You're pretty good. That's why you're the chair. So he decoded ball. He held the rule online. He selected throw, and he said it. And he did it in less than a second and a half. That's pretty remarkable. Well, it turns out if you do fMRI, you do find all the critical language areas, but it takes about 6 seconds to get the blood flow. Wernicke's area for decoding, Broca's area, motor area. If you do electrocorticography, watch what happens. This is, again, a 10-second movie, over 2 seconds. You're going to see your brain in action, and what you will see, first he heard, throw ball auditory areas, hold the rule online, frontal areas, select throw, send the message through your motor area, drive your mouth, and you refire your auditory area. So what we've done is we've completely tracked language, what you just did, in real time with direct brain recording focusing on this very high frequency band activity. It's really pretty remarkable. I remember we got this movie like in 2006, and I started looking at it. And I was in my study, and I'm looking at the movie, and my wife comes in and she says, "What are you doing?" I said, "Look at this movie." And she looks at the movie, she leaves the room, she comes back, she puts a glass of wine next to me. And I start drinking the wine, and then I stop looking at the movie. So she figured some kind of appetitive conditioning, right? So anyway. So we can track language. What can we do with it? This is work by Dean Flinker, who's an assistant professor now at NYU. He's a great grad student in the lab. And he had people doing speaking tasks, and we recorded from both Broca's area and auditory areas and motor areas that drive your mouth, the actual driver of your mouth. What he found, lo and behold, yeah, the auditory area gets massively activated when Jeff heard the word ball. And Broca's area gets activated about 150 milliseconds later, but when Jeff actually says throw, Broca's area shuts off. It's actually not active when you speak. What is active is the nearby motor cortices. This just completely disrupts the entire theory of language organization, totally gone. Now, if you then look at single trial, and this is very important, this is why we really like this frequency space, this high frequency band activity. This is single sweeps. These are 7 patients, and let's say Jeff did the first word 10 or 20, and the next patient did 10 or 20, and so on. If you see every sweep, you get high frequency band activity in Wernicke's area, and this is the actual speech onset. You can see Broca's area onsets linked to the stimulus and stops. It just stops when you begin to speak. So the actual Broca's, and this is Dean's theory, is that Broca's area is really not involved in language, it's involved in planning language. It's like a motor programming area. It's putting together the sequence that you want to send to your motor area to drive all your articulators and actually produce sound. It kind of really flips the idea of what this brain area does. And I show you this because it shows two things. This single trial activity, which when I finish the talk on how we can analyze use this activity to develop a speech prosthesis, you can see how reliable it is. It also shows this intracranial recording can address and test classic theories. How about the hippocampus? Everybody knows the hippocampus is involved in language. If you remember anything from this lecture, it will have had to gone through your hippocampus, some magic is done, and it's stored. So the words, the sound, the pictures, it all goes through your hippocampus, but nobody thinks the hippocampus is involved in actual language processing. Nobody until Victoria Paella did this study, and she's now a faculty member at Nijmegen, and she did a really simple study. She had patients from our lab who have electrodes inserted right in the hippocampus. There's the hippocampus proper. And she gave them a classic task where you say, he locked the door with there's an interval, and then they have to say as quickly as possible key, or she walked in here with and she has to say key. This is a constrained sentence. It's semantically constrained. You're heading for only one word, key, where this is an open-ended sentence. And it's been known behaviorally for years, if you have context in the sentence, this is your reaction time, you get faster naming the key in every patient, and you're slower if you have no context. And then we have some additional data that if you have a lesion, a stroke in the hippocampus, the patients don't seem to extract the fine semantic details of speech. So what did she find? Pretty simple, and that's one of the nice things about this approach. You can get pretty clear results. What she found is in every patient, each one of these is one of her 11 or 12? 12. Every patient, this is the sentence evolving from the first word to when you have to name key. As the semantic context of the sentence begins to constrain the sentence, you get rhythmic buildup of hippocampal theta in your hippocampus. So your hippocampus online is processing language. It's not a slave to the language system. It's actually part of the language system. And this again calls for a reformulation about how we understand the role of the cortex and the hippocampus in actually processing language. Again, he can't see without his. It's got to be glasses, constrained. He can't play without his. Could be anything. Again, you see this idea that this semantic constraint is dependent on the hippocampus, and she's got a lot more analysis to do on this, but she basically published this first. A really elegant work. Now, what about brain areas communicating? This is work by Ryan Canolti, and what is coherence? Not everybody knows what coherence is. It means the two things operate in sync, okay? So this is my real-world example of coherence. There's 3 kids at the playground. They're each on a swing. They want to be in sync so that when they're swinging, they can look at each other and laugh, okay? So here we go. Mom or dad pushes kid 1, and there's the kid. Here's his oscillatory pattern here. Okay, there you go. Now he's swinging. Second kid gets pushed out of sync. The third kid gets pushed. They're all out of sync at the start, but they want to get in sync because they want to communicate, and guess what? They will. It'll take them maybe 30 seconds to do it, but eventually these kids become coherent because they want to be able to look at each other. That's what your brain does, but your brain does it in 1/10 of a second. Bang. You put together brain areas, they become coherent so they can transfer information, and we can do all these amazing things that we do so rapidly. So the idea is that 2 brain areas are in sync, they can communicate better. If they're not, they can't. And this has been well determined. This is work by Ryan, and he showed that not only do brain areas become coherent, the low frequency activity, this is low frequency in the 4 hertz, that's 4 cycles per second, tunes when high frequency activity occurs. So here's a cartoon, low frequency tuning high frequency. You say, why would the brain do that? Well, the simplest reason probably is it saves a lot of energy. You think you have seamless cognition. You think everything's perfectly seamless. It's not. You're actually processing things in preconscious chunks of about 200 milliseconds. It seems to you like the world is continuous, but it's actually being brought in little frames. And this is your brain mechanism in a way for framing activity, for chunking the brain and saving energy. Your brain, by the way, is a massive energy source, a massive energy consumer. It's 3% of your body weight, but it actually consumes 20% of your cardiac output. Massive. Just huge. It's a huge energy machine. What about attention? To just show you one more example of this, here is a Starry Night task. And I'm going to show you how we have high frequency activity, coherence, and tuning of high frequency activity in this simple task. Think of it, I'm in the room, I'm looking at the camera, and I'm either be asked to pay attention to the right side of the world and detect somebody with a blue scarf on, or I'm going to be asked to detect the left side of the world. And these are 8 patients with these electrodes all over these classic brain areas that are involved in attention. I want to say all the electrodes are put in for clinical reasons. We do not have any control of where they go. They're put in based by the epileptologist, not us. So here we go. So you're targeted here, you press a button there. Next trial, you're targeted here. That's not a target because it's not in your attended hemifield, and so on. There's another attended target. So that's the task. It's beyond simple. It's a simple task, but it's a nice task because it's been used in patients with stroke and neglect to bring out their lesion, their deficits, even years after they've had a problem. So what happens? If I look, this is a patient, one patient with electrodes on their brain, and I'm going to look at the high frequency band activity. So this is the patient. They've got electrodes on the left side of their head. They're being asked to attend to the left field, right? So that's the non-attended field compared to where the grid is. What you see, they do get some high frequency band activity. There's 125 hertz. Now I simply say, pay attention to this side of the field, and what you see is a dramatic increase. So you can see this high frequency activity precisely tracks where you're paying attention. Second thing, if I say pay attention to the left field, not the right field, you look at phase coherence, how these brain areas are in sync between the frontal and parietal attention network that's well described in lesion studies and in fMRI. Blue means there's significant coherence within electrodes in the parietal cortex. Red means significant coherence between electrodes in the frontal cortex. If it's split, it means these electrodes are coherent between 2 areas, between the front and the back. So that's when you're not attending. This hemisphere is still active, but as soon as I say pay attention to the right side of the field, look what happens. You'll see many of the electrodes become split, right? What you've just done is establish coherence between 2 brain areas, so those areas can act in concert. Now, does it happen just in attention and maybe in language? It happens in everything. Here's an extremely simple study done by Ji Zhang in Jack Lin's lab, and she's very talented. They're interested in emotional regulation. And everybody in this room, well, I'm sorry, not everybody in this room, but it's well known in the literature that the amygdala is a very important structure in the brain for decoding the emotional valence of the world. It's like your emotional sensory part of your brain. And it's also known that if something emotional happens, you're more likely to remember it, right? So you get a memory boost. So how does that work? What's the physiology of this? Can we understand it? And maybe can we eventually regulate it in neurological and psychiatric disorders? So here's your amygdala. Here's your hippocampus. Here's electrodes in our patients, either in the amygdala or in the hippocampus. And this just came out. It's a really nice paper. She showed the patients either landscapes, which are neutral, of course, that could be where a serial killer dumps bodies, I guess. I don't know. If they're different people, it might mean different things. And fearful faces, but these are really fearful. That's the best, I think. In any event, so the patients are, this is pretty simple. They're just looking at movies. But it's naturalistic, and it really drives the brain. What's the results? The results are really straightforward, very clear. Activity, theta band activity, low frequency activity in the amygdala drives high-frequency activity in the hippocampus. So this is your brain, and when you remember something that's emotional, this is what's going on. Your amygdala extracted that it was emotional, and then it sent a signal to your hippocampus to say, hey, maybe we want to remember this thing. So this is really interesting because this kind of control of emotional processing, as many people know, is abnormal in many psychiatric disorders. So finally, in this part of the talk, and then I'll have another, I'll be on time. I put this in the talk because it just got accepted this morning after 2 years of misery with the reviewers. So all of you out there having trouble with reviewers, guess what? It's normal. Just deal with it. It's harder the younger you are because your time frame is shorter, so you want to get your PhD stuff out. I just go, whatever. Let's do whatever they say and pray that they'll accept the paper. This is work by Matar Haller, who was a grad student in the lab, and Augusta Shestiuk, who was a postdoc in the lab. They're both very, very talented. They did a series of tasks, from very easy tasks like categorization, dog versus cat, to what gender is the person, to repeat a word, to auditory categorization, male or female voice, to very hard stuff, antonym. I say good, you come up with bad. And you can see the tasks get harder and harder because this is the reaction time, easy to hard. This is a huge study. She had 16 subjects, 8 tasks, 38 recording sessions. This is a lot of data. One of the first reviewers said, well, why don't you do this in fMRI? And I resisted my urge to reach through the computer and strangle them. But anyway, was that wrong too? Bad? Can I say that in Canada? It's normal. You just don't say it in Canada. We say it. Anyway, so what did they show? This is incredible. They found how the brain links perception and action. Perception is doing all these studies, and in each one of those you had to press a button. So what do you see? Tons of electrodes. These are our active electrodes as determined by high-frequency band activity. I just modified this today from in revision to impress, so the two of them are ecstatic. What you find are 4 kinds of electrodes in your brain across all these tasks. Electrodes that are linked to the sensory stimulus, electrodes that are linked to the response when you press the button. This is the gray is the offset of stimuli, and electrodes that link the perception and the response. And these electrodes are centered in the frontal lobe, and these are the really interesting ones. We've known about these for a long time. Everybody in the motor electrodes, what am I doing here? Go back. Okay, we know a lot about the motor system. We know a lot about the sensory system. What we want to know is how we bridge perception and action. The results are just, I think really incredible. I shall skip this. This is just how these networks go up and down together on a single trial. The sensory and the response and the linking electrodes in the frontal lobe go up and down together in terms of their power. What they found is that if you look at the activity, this is across all those different tests. It's task independent. This data is sorted just by how fast you responded. So there could be a fast response in a tough task, or a slow response in an easy task. So they're just sorted by reaction time. What you see is the sustained activity in the frontal lobe. This red thing goes up. Here's the reaction time. Here's the blue line is the activity in the motor cortices. And if you look, this divergence point between the frontal cortex and the motor cortex here, precisely predicts when you're going to press the button. We have a linkage between the frontal lobe has taken the perceptual information, held it online, formulated some decision to press a button, and handed off the information to the motor system. Statistics are embarrassing. The effect of this point of divergence, predicting how fast you will respond, is a p 10 to the minus 11th. That's a big effect. That's a huge effect. So anyway, they're happy, I'm happy. They're happier than me. But actually, I'm happy that they're happy. So what about a speech prosthetic device? Well, we're going to close with that. So I'm giving you little pieces of stuff we're going in the lab. I don't think it's the most organized, one topic talk, but I wanted to give you a feel for how you can do brain recording to kind of use it to give you insights into behavior. So speech prosthesis. Stephen Hawking, that should be no G. ALS, terrible disease. You saw the aphasic person that couldn't speak. Minimally conscious state, developmental populations. Can we, when someone wants to speak, when Stephen Hawking wants to speak, but he can't because he has this horrible neuromuscular disease, could we decode his thought? Okay. And could he speak through a computer? People said we were bonkers to even try. And we still may be, but let me show you where we're at. This is an electrode placed right on your auditory area of the brain, and some of these recordings are done in the operating room. Okay. So this is a patient who's awake, about to get surgery, and we're mapping them in the operating room. For those fMRI people, that's your cardioballistic. Every time your heart beats, your brain pulses. And then we do Penfield mapping of Canadian, brilliant, McGill. He started this whole field in the 40s and 50s.
We're going to get ready to do the
When
mapping.
he's finished mapping, we put a high-density grid on if the patient's up for it. Sometimes they just want to proceed with the surgery. And we record directly from their brain. So the first thing, if you look at these 9 electrodes, you can see these ones on the outside have activity when you hear a word. The electrode here does not respond when you hear, it only responds when you speak. So the first thing it tells you, this idea that there's a blob of language cortex is ridiculous. There's an exquisite microstructure, at least at the 4 millimeter domain, and I think it's actually 2 millimeters. Okay, so that's number 1. And you can see high frequency to hearing, single trial, high frequency to speaking. So this was exciting. We now have even higher density grids that we put on, and this is a 250 channel grid, and this is just recording of phonemes in the temporal lobe of humans. Okay, so where's this going? We had to figure out really what's going on in your auditory system. How fast does it work? How plastic is it? Before we got into a prosthetic. So we first tackled categorical perception. Categorical perception was reported in 1956. You're going to hear a sound that just changes perfectly linearly over 14 steps, but that's not what you're going to hear. You're going to hear 3 sounds. You're going to chunk it into bas, das, and gas.
La, 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 ga, 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 ga.
Did you perceive that? You didn't hear continuous 14 sounds. You heard 3. You bucketed it. This is another mechanism that we use all the time to save energy. We don't have to take, so the question is, do we take everything in and then bucket it into the 3 sounds, or do we bucket it automatically and quickly? This is very nice work by Eddie Chang and Joachim Rieger. The answer, actually, using direct brain recording, is that we chunk it immediately within 120 milliseconds. We actually split up the sounds in our auditory regions into bas, das, and gas. And you read, the paper has all the methods. And by the way, if anybody wants to get into this area, everything we have, every analytic software we have is available to anybody who wants it. So if you start doing this kind of research, contact our lab, you can have whatever you want. Basically, so this was interesting. This gave us, we're starting to get a little grasp on what's going on in auditory areas. And then Chris did this study, Chris Holgraf, just finished, just got his PhD. And what we did is we took speech phrases and we extracted the language component of the speech. And it sounds like garbledygook when you hear it. Then we played the right phrase, the real phrase, without the language component taken out. Then we played the garbled stuff again, and I know what you're going to hear. You're going to hear garbage, you're going to hear the perfect sound, and then within a second, the garbage is going to turn into the real sound. This highlights the incredible sub-second plasticity of your brain.
Bagpipes and bongos are musical instruments.
Bagpipes and bongos are musical instruments.
Bagpipes and bongos are musical instruments.
That's the same noise.
The high security prison was surrounded by barbed wire.
The high-security prison was surrounded by barbed wire.
The high security prison was surrounded by barbed wire.
You perceive it?
The full moon shone brightly that night.
It's crazy.
Move the garbage nearer to the large window.
So I think you believe it's a real phenomena, and it turns out what's happening, your auditory cortex, ensembles of cells that were not language responsive, switch with one exposure to language to being language responsive rapidly. This is just, really pretty amazing. And you can now, this is the behaviorals, you can now pick out what words were said here. If I have no context like white noise or pink noise, there's no behavioral effect. It's not just repeating these phrases. If I have the wrong context, the wrong sentence, I get a teeny behavioral effect, but nothing like the right context. So this was pretty exciting. And I won't go into the details of this in the sake of time, but basically the simple thing is the tuning function of these cells in your auditory area switch from non-language to language in a second. So now we're getting excited. So the next thing is to do speech reconstruction. Can we reconstruct your speech when I say something to you? This is work done by Brian Paisley, and I'm going to give some advice to the faculty here. When your postdoc or grad student, not your grad student, no. When your postdoc comes to you with a crazy idea and you think it's not going to work, do it anyway. He came to me and said, there's a lot of people doing motor BMI, right, controlling a wheelchair, and it's not simple, but it's relatively simple because you just need to be able to have a signal to go left, go right from your brain. We can do that. He said, I want to decode speech. I'm going, oh, great, that'll be interesting. But we did it. He did it. Actually, yeah. So we presented 100 words to patients with these high-density electrodes in auditory areas. Some of them were epilepsy patients with high-density grids. Some were intra-op patients. We recorded their brain electrical activity, the electrocorticogram, which I've shown you, the tremendous signal you have in here. We focused on the high-frequency band. We came up with a reconstruction model to see if we could take this electrical activity and turn it back into, let's say, baseball. Well, it's Canada, hockey. And it turns out that you can. But, modeling has a lot of parameters, and you can't just believe your model, you have to test your model. So Brian kept out of the data. He created the model with about 70 of the words, and then tested the model by presenting the 30 words to the model to see if the model could distinguish if it was hockey or baseball. Chances 50%, he got 91.8% accuracy. I almost fell over. What he's doing, and this is just in frequency analysis, we're now doing phoneme and semantic and deep neural learning and all that, maybe in a couple years we can tell you even more. What he's doing is reading the notes of the cortex. Beethoven's deaf. If he looks at someone playing was deaf. If he looks at someone playing piano keys, he can reconstruct in his mind what's being played. That's what we're doing. Each electrode you can think of as a specific piano key being hit at a specific time and generating a specific frequency. Okay? We're trying to read the notes of the cortex. So 92%, what does that mean? It's mathematically highly significant. I said, Brian, make a sound file. Brian said to me, it's not scientific. I said, do you want to get a job? He said, I got it. So it's fun having a lab. No one pays attention to me until the very end. And then it's just a teeny bit. So it's kind of fun. So here, what you're going to hear are words that the person heard, and the reconstruction of the word played directly from brain electrical recording. Nothing else in between. So here we go. either these high density in surgery or high density epilepsy grids.
Waldo? Structure. Doubt. Property.
Not bad, right? And that's the first shot out of the box in 2012, and it's like your first love. You got to show that one. We're better than that. So, but it's really been exciting. So we can decode what you've heard. That has no help for the patient at all. We can only help the patient if we can decode what they imagine, right? That's what we need to get at. We need to know when Stephen Hawking wants to give us another theory of how everything works. Oh, we can do single phrases from the brain.
That's my fire escape.
We've reconstructed that phrase from direct electrical recording. These are just the reconstructed, the real, the heard, and the reconstructed spectrogram. Okay, so how can we get at imagine? Imagine's hard because everybody, if I ask you to imagine a word, everybody imagines it a little bit different latency, a little bit different length. So we needed some way to track this. So the way we tracked it is, does everybody know what the Gettysburg Address is? The Americans need to. This is the famous Abraham Lincoln speech. We chose that because we could play a ticker tape to the patients. And in one condition, they had, so the ticker tape goes by, and in one condition, the patient repeats the words. In another condition, whoops, they imagine the word.
47 years.
I have those out of sync, but you can get the idea. So now we have a code. We have the ticker tape where we can look for imagined activity, right? We have some kind of idea of when we think the patient is at least imagining.
Consonant.
We also have studies now that we're doing in the operating room and in the epilepsy patients where we have a more controlled one where we have an auditory cue. They hear a word, let's say hockey. They get a visual, let's say Edmonton Oilers. They get an auditory cue, then they get a visual cue and they have to imagine what they just heard. Then they get another cue where they have to say it. So we know we can decode. I just showed you we can decode when they hear. You'll have to believe me, I can decode. If you say baseball and you hear it, I can decode your auditory cortex having heard baseball. We have two models to compare to. So we have this data set and this data set, and to cut to the chase, we can reconstruct when you imagine. And this is work done by Stephanie Martin, who's just got her PhD in EPFL. She did a joint PhD between Berkeley and an EPFL in Geneva. And basically, this is the kind of data I showed you where I can decode what you've heard. If I say baseball, I can decode baseball and play it back. And this is decoding imagination. Now, it is not as robust, but it's highly significant in every patient. So we're very excited that we're in the right track. The informative electrodes are, again, as one might expect, predominantly in the superior temporal regions of the brain, these key auditory regions. Here, here, and here. These are the electrodes providing information, for the decoding. So we can use this where we can do categorical decoding, so we can have the patient pick between 6 categories, like I'm hungry, I love you, What we can't do yet is get it to the point where we can decode anything. We're not quite there yet. And I don't know if we'll ever get there, but we're not going to stop until we get there. Did that make sense? I don't know. It's really important because this would be very valuable for patients. And there's nothing better than doing something that really can help somebody. So what about music? I'll conclude with this. Who likes Pink Floyd? How many people like Pink Floyd? Everybody? Does everybody know Another Brick in the Wall? Who knows Another Brick in the Wall? Yeah? Okay, great. What's wrong with this album? What is wrong with this album if I'm going to decode Another Brick in the Wall? It's the wrong album cover. I think it's The Other Side of the Moon, right? The Dark Side of the Moon. The reason I use this, again, is because it shows frequency decoding, and I thought it matches what we're doing, but congratulations, not everybody knows that very good. So here's what the patients hear. We let the patients do whatever they want. They can pick any music, play their own stuff, and a lot of them, over time, listen to Pink Floyd.
Just a break in the wall.
This has been turned into a nursery rhyme in Brazil, actually. It's so universally loved. The greats. Okay, so the patients listen to this. Question is, can we decode Pink Floyd from the brain and play it back? Okay, this is work done by Ludovic Bellier. He's a French postdoc. We had lots of electrodes across 28 patients. These are the electrodes that led to effective decoding. Most are in the left hemisphere, some are in the right. I want you to notice there's also ventral premotor electrodes, but they tend to be in the areas that I've shown you are critical for auditory processing. So the song is 2 minutes long, and what Ludovic did is take the first minute and come up with a decoding model. Just like I told you, Brian's work, taking some words, making a decoding model, and then seeing if he'd decode the words that the model had never heard before. So here's the results. This is what the patient heard. I've cut it at 7 kilohertz. So it sounds a little grainy. It's at 7 kilohertz mainly because of the computational problems. If we go much higher, it takes a couple days to run some of these algorithms. And if we go to 14 kilohertz, it's taken a week. So here's what the patient heard. Just a second here. Cover another brick in the wall. Whistle, words, sound. Okay, here's Pink Floyd played back from your brain. I am very hesitant to publish this paper because I know what's going to happen. People are going to, that's the only thing they'll ever remember we ever did. Oh, that's the Pink Floyd group. Pink Floyd played back, decoded from the brain. So we can decode language, we can decode music. Music is important because it has prosody. And if you want an effective decoder, it would be nice to know if the patient is communicating with affect, a neutral I love you, real I love you, or affect. We can begin to decode imagined speech, and then finally we can decode imagined music. And this paper just came out 2 weeks ago. This is a wonderful young guy who contacted us with his dad. And I can talk about him because he's a co-author on the paper. He was 16 when he contacted us, 16 or 15. With his dad, he said, I know you guys are doing stuff with music and brain reconstruction. I'm going to get surgery. I've got epilepsy. I've got some kind of epileptic source in my language area. And I'm going to UCSF. Can I participate in research? So we said, sure. And he spent about 6 weeks learning 2 Brahms and Beethoven new pieces and on his keyboard. And in one condition, he played the keyboard and he heard the sounds. So we could decode the sounds. In another condition, he played the keyboard and he imagined the sounds. So, but the beauty of that is we had a way to look for imagine. We had a timing. Really simple. The funny thing about this is that he ended up in my freshman seminar at Berkeley this year. He's actually a student at Berkeley. The surgery was highly successful. He's doing absolutely great. Super nice kid. And basically, this is, again, music imagination. Again, just came out. Stephanie Martin. And we have electrodes that respond both to when he played the music and when he imagined the music. And we have electrodes that respond to only when he played the music. And I won't go into all the details of this for the sake of time. But if there's any questions about this, please let us know. And here's the actual electrode grid over his auditory area. Here's activations when he perceives the music that he's playing. He's hearing the music, and I've already shown you lots of these activations. Just a little blow up an area here. Here's the activation in his brain when he imagines the music. It's in the same locations. And and you can see it's not as robust, but it's definitely there. We're very excited about this because now we've been able to decode Imagine speech and imagine music. It's set the infrastructure for the next wave of research, which is going to require higher density grids. We need 2 millimeter grids. We've got to get them built. No one wants to make them because they're not, anyway. We need better computational approaches, but we think we're beginning to get traction. The goal is we will be able to take a patient like Stephen Hawking or a patient like that gentleman with Broca's aphasia and do a small burr hole, put an electrode in on a strip that we fan out that covers the auditory areas, and we basically decode when they want to speak to you. And they can we can restore speech. That's the goal of the project. We're talking with a group in Germany which has 4 very unfortunate people in their 20s with very bad ALS. They're completely locked in, and we may implant them. The patients want to do it, but there's some logistics between the U.S. and Germany, and we'll see. So I'll stop with. I want to thank some of the people. I presented the data from, and the starred ones I presented data from. The non-starred ones I didn't. I just want to highlight one of your grad students, Julia Kim, who has fabulous data that she's been working on neural networks for mind wandering, and I'll wait another time to present that or let her present it. But they're really a great group, and I just want to point out that there should be no borders to science. Israel, U.S., France, U.S., Israel, U.S., Netherlands, Ukraine, China, France, Israel, Netherlands, U.S., Germany, Canada, U.S., Germany, and so on. Switzerland, U.S., Netherlands, Iran, and Germany. So it's an international, the students, talent is everywhere. And I think for the sake of time, I'll stop right there. And I want to really thank you. I know some of this is out of people's normal daily consumption, but thank you for your attention. Happy to take questions. Any complaints, Jeff?
I think we have a couple microphones.
Sure.
On either side of the room, so do you want to come out to the speaker? Go in.
It was so clear no one has a question?
Bob?
That's fine.
Bob, I'll ask a question.
I don't take questions from faculty. Go ahead.
I was just wondering about I'm going to call it an illusion, where you hear the speech and then you get what sound like gobbledygook, and then
turns into you hear it. Yeah.
How much in your examples afterwards was that actually, do you think, happening to us, where we were hearing Pink Floyd, having heard Pink Floyd, and so forth? Like, would we have heard the reconstruction clearly if we hadn't been exposed?
Yeah, I think there's always a priming effect, but when you do some of the behavioral stuff, when we ask people to rate them, for purposes of this presentation, you put them there, but you counterbalance them to which it is. You kind of present the degraded before the actual degraded. The other thing, if you remember, one of the slides in there, the stuff when the tuning things were changing with Chris Holgroff's data, we have lots of controls. So in between the two presentations, there's pink noise or white noise, so it's not a repetition effect. We have different language in between that doesn't help. So we try to control for those things. But it's a great, what you do is you take the data that you get from the patients, and then you have undergrads who rate them with some counterbalance. That's the only way to not get away from the idea that I'm priming. I am priming everybody to get the effect because I want you to hear the effect, but the effect is definitely real. Yes, ma'am.
Such a great talk. One
Thank
of
you.
the best talks I've ever heard. Thank you.
Thank you. Can you talk to my mother about that?
Absolutely.
Okay, good. You have to pray to get to her because she's
Well,
not
I
here.
can do that.
Okay, thank you.
So I'm not quite sure where to start, but I think I'll start here. It's kind of follows up a little bit from Alan's question. So you have the human brain. You have a perceiver who is designed, has learned to deal with this type of information, and you have a certain level of expertise of the imaginer. So you couldn't have just anybody imagine that music. He was obviously already a pretty talented pianist.
Absolutely.
Yeah.
Absolutely. That's why it was great to get access
That's
to
why
him.
he was great. And we're all fairly talented in language.
Yeah.
But I'm just wondering, now I do a lot of work with audiovisual speech, and one of the things we're really interested in is we can also, well, this was Eric Bateson-Bateson's work, you can reconstruct the acoustic signal from visual facial. And you can reconstruct with a robotic visual facial thing, the visual facial movements from the acoustic signal. So how much of it has to do with the relationship between sources and perceivers that are designed to or have a lot of experience with that particular sharing of information, or could we reconstruct this, from signals bouncing off a wall? Is there is it?
You could bounce any of the sound file off the wall, and if you had the right acoustic pickup, you could decode it.
Right, and
sure.
then turn it back into something.
And turn it back into the sound file. So you bring a couple really important points. The first thing is that we get almost, the people who've been participating in this research they've not been trained on the paradigm. We don't get them because they come into the hospital, let's say at 12 on Tuesday, and they're going to surgery Wednesday morning. All we have with them is maybe a 20-minute chunk. So what you brought up is okay, let me back up. If you take a cochlear implant and someone who can't hear, right, they've lost 25,000 hair cells, you put a cochlear implant in, and it's got 64 stimulation channels. When you first put it in, it sounds to the receiver like gobbledygook. What the brain does over a month, it takes those 64 channels and turns it into decoded speech. So if you've hit one of our key problems. The key problem is we'd like to be able to train the patients on the task for a month or two before they went into surgery. I think we would get much better. I think we'd get much better results. That's why I think if we had these electrodes implanted in someone with ALS, who has every minute of the day they want to be able to speak, I mean, I can't prove this to you, but I think we'd accelerate the research because of that exact problem. The kid was a pro, right? He's a pro. So it's just a case report that shows it's feasible. But the real problem is we don't. The rest of all the data from everybody else you've seen, walk into the room, will you listen to this and do this? 20 minutes done, and we're out. So there's a problem. It's not a problem. It's just a clinical issue. It's a coordination problem. And one of the problems for me is I moved from the medical school to Berkeley, which was great because of a lot of things at Berkeley are wonderful. But it's not a medical school, so I can't control everything. If I was at a medical school, I'd have it set up. I'd know when the patients were coming. I'd have someone go to their house. I'd have them work with them for a month before we even tested them. And I think we would get better. But, we do a lot of this auditory reconstruction at Albany Medical Center. And we just got a large Conti grant from the NIMH, and part of it is this reconstruction. And we're actually now talking with the epilepsy people about doing just what you suggested, getting a research technician to go to the patient's house and work with them, training them on the imagination paradigm so that we don't walk into the room cold and have to kind of hope that something's going to work. But that's a great point, for sure. Yes, please.
You showed earlier that low-frequency oscillations in the amygdala drive high-frequency oscillations in the hippocampus.
Yeah.
Are you able to also add electrodes in a third area, such as the prefrontal cortex, to see how the prefrontal cortex is able to regulate this the emotions? So to look at this whole circuit.
The answer is yes. We have the data for orbital frontal and lateral frontal cortex. So you're spot on. I mean, that's exactly what we're looking at. When you're doing this kind of work with these complex data sets, you got multiple brain areas, multiple frequency bands. If you try to solve the whole puzzle, you start to go crazy. So we take off chunks, get control, and build on it. So the stuff I showed you with the hippocampus and theta being involved in speech, we have electrodes all over the lateral language areas. And what Victoria's doing right now is doing just what you're suggesting. She's looking at the interaction between the auditory cortical areas and the hippocampus. Similarly for the emotion, we know orbital frontal cortex is a key part of this particular network. So it all comes down to anatomy, physiology, behavior, and realizing the brain is not a spot, it's a network. So that's another great question. Thank you very much.
Really exciting. Thank you.
Thank you. You need an epilepsy program in Vancouver. Canada needs one. You started this whole field with Penfield and McGill, and McGill doesn't have an epilepsy a research program. I'm sorry, I shouldn't tell you what you need. Actually, yeah, I do. You need one. Okay, please.
Can you elaborate a little bit on how you do the acoustic reconstruction? And the follow-up to that, have you considered using machine learning?
We do use
machine
learning. That's what we use. That's exactly correct. We use machine learning, and we do multiple regressions on the different electrodes, trying to relate them to different acoustic features. We do machine learning, and we've just, like discussed with some of your colleagues here, we don't have enough data to apply deep neural networks yet. You need a lot more data than 20 minutes. So what we've just got IRB approval for is that we can record 24 hours a day in the patient's room. So every time they talk with anybody, we will have the data. And hopefully that'll allow us to go to the next level of reconstruction. We're already doing overnight sleep, and the sleep turns out to be extremely interesting. I mean, I obviously didn't have time to talk about it today, but you're right. I mean, these are multiple regressions, machine learning, all these mathematical techniques are what you need, because you have not only the multiple electrode spots. When you're looking at the network stuff, you have multiple electrodes and multiple interactions. So I didn't really talk about it, but the reconstruction is actually, there's a top-down signal from the frontal cortex in the theta band that regulates the auditory areas during language perception. So it's even more, I kind of just showed you a piece of the story. It's a complicated story, basically, but I think it's tractable, for sure. Thank you. Yeah. Okay, what I'm going to do, can I do 2 minutes? Do you got 2 more minutes in your brain? I want to talk to you about a kid's journal real quickly, because I think you guys I had a wonderful time today, and I think you'd be great to be engaged in this. We started a kids journal called Frontiers for Young Minds, where kids 8 to 14 review papers. They're papers submitted by scientists. They're targeted for kids 8 to 12. Okay? They have 3 pages, 3 references, 3 figures. The paper, they come from scientists, right? And we have 6 fields: neuroscience, biodiversity, earth, space, health, and now mathematics. We're going to add robotics. Scientists write a paper, you submit it to the kids' journal. It could be a new discovery, fuel from plants, how do we see it? We'll take either kind of paper. The paper is then sent to a kid.
The kid is hooked up with a mentor who could be a scientist in their family, but more commonly is a PhD student or a postdoc. They review the paper. They then formulate a review. It's just like the standard review process. What we want to do is we want kids to understand at an early age the scientific process. How do you formulate an idea, hypothesis? How do you design an experiment? What's data collection? What's data analysis? How do you write it up? And how do you get it published? Okay, so the researcher, most of the mentors, it could be anybody in this room. You want to get involved, great. The kids provide a constructive review, and the kids feel empowered because the scientist has to do whatever the kid wants. Okay? I'm the chief editor. There's been 150 publications. There have been no appeals. You do what the kid wants or you take a hike. And what it does, it assures that it gets the kids excited because they're empowered, but it also makes sure we have a corpus of papers that are understandable by 10 or 11 or 12-year-olds. Every article gets a cartoon, okay? So you can see here, I think you could figure out what these articles are about. The top left one is there life in other planets? The one on the top right is, extraction of carbon dioxide. The one on the left is using stem cells for medicine. The one on the right is wear a helmet. You're going to get a concussion. And then if you go look at the website, Frontiers for Young Minds, the kids, we don't publish the kid's name or face. They put an avatar up. They can make their avatar or send them an avatar program. And they put their name, their first name only. You click on their avatar and they have their bio that they wrote. It's on the face page of the journal. And the kids are brutal. All the niceties, well, the adult review process is snotty, but it's not really snotty. The kids are just cute because they say what they think. So let's take a look at some of the reviews. It would be helpful if they told us how they took the measurements of brains without actually having to remove the brain. That's a reasonable question. How about this one? The writers of the article did not make it clear why such an expensive and involved research project was done to begin with. It seemed like a fruitless task. That's called ouch. It's a big ouch. This was an article on fMRI of reward and Facebook usage. And the kid, I remember the kid, in his review, he said, the reward's 80 cents. My friends don't care about 80 cents. and he said, and I have no idea why they did this. It's not going to help anybody. How about this kid? The point is not clearly, this is a 10-year-old. I didn't understand the main scientific question because there were so many details at the beginning. Clean up your intros. Maybe state what the main question is earlier in the manuscript. And finally, the dreaded review. This is the one you don't want. This is the one you really don't want. This seems important, but the way it is written is so boring, I can't even get to the end. Could the authors maybe sound excited about what they are doing? So if you want to have a good time, get involved with this journal because the kids are absolutely fantastic. Thank you very much for your attention.